Now we'll look at how motility is generated in the GI tract. Motility is the contraction and relaxation of the smooth muscle of the GI tract, and it determines the flow of the luminal contents. So as we saw, the GI tract is made up of layers of the mucosa that's in contact with the luminal contents, the submucosa, the muscularis externa, and the outer serosa. It's the inner circular smooth muscle and the outer longitudinal smooth muscle of the muscularis externa that are responsible for motility in the GI tract. When the circular muscle contracts, it results in a decrease in the diameter of the tube at that point. And when the longitudinal smooth muscle contracts, it results in a shortening of the tube, decreasing the length of that segment. The enteric nervous system is also important in regulating contraction of the gastrointestinal smooth muscle, and it's primarily the myenteric plexus that controls motility, while the submucosal plexus regulates gastrointestinal secretions. Another important factor in generating gastrointestinal motility are the interstitial cells of Cajal. These interstitial cells of Cajal are specialized smooth muscle cells that are broadly distributed in networks throughout the gastrointestinal tract. They're considered to be electrical pacemakers for smooth muscles of the GI tract, and they're thought to generate slow waves that look like this. Slow waves are spontaneous, cyclic depolarizations and repolarizations of the membrane potential that are then able to spread through the smooth muscle by way of gap junctions. During the depolarization phase, there's an influx of calcium. The calcium current slowly inactivates and is balanced by an outward potassium current, and eventually the potassium current dominates, leading to repolarization. This continual, slow, intrinsic electrical activity spreads from the interstitial cells of Cajal through the smooth muscle via these gap junctions. And the frequency of the slow waves generated by the interstitial cells of Cajal varies along the gastrointestinal tract. So the stomach has only about 3 slow waves per minute, while the duodenum has more like 12 slow waves per minute. If at the plateau of the slow wave, the membrane potential of the smooth muscle cell is depolarized to this threshold, then that's when action potentials or spike potentials occur. The higher the slow wave potential rises, the greater the frequency of the spike potentials. Contraction then follows that electrical response. So slow waves themselves are not action potentials, but are just oscillating depolarization and repolarizations. And by themselves, slow waves don't cause strong muscle contractions. It's only when the depolarization of the slow wave exceeds the threshold that an action potential occurs. So it is the frequency of the slow waves that determines the frequency of possible action potentials and therefore the number of possible contractions per minute. This diagram shows the slow waves created by the interstitial cells of Cajal and the spike potentials or action potentials that can occur. So here at rest, even sub-threshold slow waves do produce a baseline tension or tone, and the smooth muscle is not completely relaxed. This tone is altered by neurotransmitters, hormones, paracrine substances, and drugs, and it's particularly important in the sphincters and in regions where storage is an important function. Upon stimulation, the extent of depolarization and the frequency of action potentials are enhanced. So the greater the number of action potentials that occur at the peak of that slow wave, the more intense the contraction of the smooth muscle is. Inhibitory agents lower the amplitude of that plateau phase and decrease the muscle tone and the likelihood of smooth muscle contraction. In the myenteric plexus, excitatory motor neurons stimulate smooth muscle contraction primarily through the release of acetylcholine, and inhibitory motor neurons stimulate relaxation primarily through release of nitric oxide. There are a few general patterns of motility that we'll keep coming back to, and the first is peristalsis. So peristaltic contractions are propulsive, propagated contractions that propel material along the GI tract. This is a moving ring of contraction that's found throughout the GI tract in regions that are concerned with the forward movement. There's contraction of the circular muscle in the propulsive segment behind the bolus, and contraction of the longitudinal muscle in the receiving segment in front of the bolus. Peristaltic activity can be increased or decreased by autonomic activity, but its occurrence is completely independent of extrinsic innervation. Trituration is the process of crushing and grinding ingested food by the stomach, and it decreases the particle size and increases the surface area to improve the efficiency of digestive enzymes in the stomach. Segmental contractions are mixing contractions in a local area that don't promote forward movement. So during segmentation, there are alternating contractions of the circular muscles to divide and mix those luminal contents. This type of movement is common in the small intestine. The stomach and colon also have reservoir functions, and the smooth muscle has to be relaxed to hold the contents that are delivered. The stomach holds ingested food for slow delivery into the duodenum, and the colon holds the material and absorbs excess water until defecation is convenient.